Hi, everybody. We're Team 15 and we created a bioengineered stent. Heart disease is the leading cause of death in the world, with coronary artery disease being number one within heart disease, with 17.8 million deaths annually in the world. Stents are used to treat coronary artery disease. This is a disease where there is a plaque buildup due to numerous causes like smoking, diabetes, the type of food you're eating, and more. This is seen as a global problem, and more cases are seen in urban areas because the causes are more available there. Restenosis is when the artery starts to narrow again after the stent has been implanted. Over 2 million stents are implanted in the US every year, and in just under a quarter of that amount, restenosis occurs.
So bare metal stents are considered the first generation stents and have a high probability of restenosis and thrombosis occurring, up to 41%. So drug-eluting stents are considered the second generation, and these stents have a coating of a drug on the outside to help the stent integrate into the body easier. There's still a 7 to 13% chance that restenosis occurs in these stents, however. Anti-proliferative drugs have been introduced into these systems to reduce restenosis rates. However, although reducing the risk, they haven't solved the problem. The third generation of stents would be epithelial cell recruiting stents and would allow the stent to very easily integrate into the body by taking the patient's own cells to coat the stent. So currently there are no FDA-approved stents on the market. However, we can look at some of our competition. Boston Scientific is one of the leading competitors in the industry and has a very strong foothold in the drug-eluting stent market. Their Synergy line is the only drug-eluting stent approved with a bioabsorbable polymer. And then Medtronic also has a very strong foothold in the industry. And looking at their Resolute line, they are flexible and a single wire design, making them very flexible.
Next, we're going to look at the engineering criteria needed. So for the mechanical needs, the size, the intimal radius needs to be about 10% larger than a healthy artery. And as for the implantation, the inflation pressure needed for the deployment needs to equal the material deformation pressure plus the needed stent pressure. And we also need to mitigate the stress that the stent applies to the environment, as it can lead to higher rates of plaque fraction and other biological responses that could potentially lead to restenosis. And the stent also needs to be rigid enough to withstand the stresses, as the intimal elastic lamina can have upwards to 6 3/4 greater stress than a healthy artery. And we also need to have a surface that has coating adherence capabilities. As for the biological needs, we cannot disrupt the cell function, and we need to create an environment that encourages endothelial repair. So we'll be targeting epithelial progenitor cells. And we also need to mitigate inflammatory responses, because inflammatory responses causes biological cascades that can lead to things like thrombosis and infection. And we also need to have non-cytotoxic and non-erodible materials. And because we're using heparin, the elution of drugs needs to be regulated and easily controlled. And now we're going to talk about the biomaterials. We chose a 316L stainless steel for the body, as it is used a lot in cardiac stents. And then we'll be using CD30/40 antibodies to recruit the epithelial progenitor cells and the epithelial cells as well. And the heparin and collagen complex are going to be used to immobilize the CD34 antibodies. And heparin's an anticoagulant, which will help with thrombosis. And the heparin and collagen complex is thromboresistant. And collagen has the ability to for the pressure of the arterial walls, and also promotes endothelial growth. And then we'll also be using osteopotent, which is an integrin.
So our solution is the creation of a multi-layer coating of CD34 antibodies along with a heparin collagen multilayer, which in turn will immobilize the antibodies binding to vascular endothelial cells. Osteopotent is again a multifunctional protein with adhesive functions that will be used to increase specific binding to vascular ECs. In vivo experiments indicate that CD34 antibody can enrich and accelerate the attachment of vascular cells onto stents with rapid endothelialization. The most current approach is to coat antibodies on a stent surface to attract circulating EPCs to the stent which can then differentiate into endothelial-like cells. This can be done by stimulating the neighboring endothelial cells or capturing circulating cells directly from blood circulation. A CD34 antibody functionalized multilayer of heparin and collagen is seen here layer by layer. Using CD34 antibody, functionalized heparin and collagen multilayers can specifically promote the attachment and growth of vascular ECs. Collagen itself has weak antigenicity, which makes it a very strong candidate for stent coatings. Heparin itself as well has useful effects in preventing intimal hyperplasia and inhibiting smooth muscle cell proliferation and migration. Immunofluorescence microscopy confirmed that EPCs express platelet endothelial cell adhesion. When EPCs were cultured on osteopotin-coated slides, alpha-V integrin subunit was observed in focal adhesions at the basal surfaces of EPCs. Osteopontin binds to the alpha-V integrin subunit on EPCs to support EPC adhesion and increase incorporation into angiogenic vascular networks.
The stent would be fabricated using a microelectric discharge machining. This is a process where the metal is removed using dielectric fluid and ions. The battery provides a negative charge to the tool and a positive charge to the stent. The process of ionization takes place with the tool releasing electrons and the metal ions going back towards the workpiece. Balloon angioplasty is used to inflate the stent once it is processed and placed in the patient. Using the micro-EDM leaves sharp and rough surfaces. To create a smooth surface, electropolishing is used. The piece is holding the stent in an anode and is placed into an acid bath. A cathode is placed inside the bath as well, and when a current is applied, the stent forms bubbles coming off the surface, indicating that the surface is polishing. This process smooths the surface of the stent and creates a shine. Dipping the stent does not get the tight crevices. The most popular way to coat stents is using spray nozzles. A low-pressure gas stream is used to make sure that small droplets come out in a narrow line. The coating is taken through the side inlet, while air is coming through the top inlet, pushing the coating down towards the nozzle. Ultrasonic spray nozzles give an even coat of the coating. For our stent, we plan on using ethylene oxide sterilization. This is used by 50% of the terminal sterilization market. The process of sterilization includes placing the stent in a sealed, breathable package and placing it within the chamber. Pictured as a very large chamber that can hold 2 pallets worth of products. The packaging allows ethylene oxide to move in and out of the bag as well as humidity, but keeps bacteria out. The humidity kills the microbes inside the bag. The cycle time between increasing ethylene oxide in the chamber and making sure that there is little ethylene oxide before a human approaches takes between 6 hours to several days.
Now we're going to talk about the biocompatibility and the functional testing. So hemolysis assessment, in accordance with ISO standard 10993 section 4. It measures the hemocompatibility and hematocompatibility. Cytotoxicity can be measured through the ISO elution in accordance to ISO standard 10993 section 5 and can be classified using visual grading or quantitative assessment. And as for the systemic toxicity, we'll be using animal testing, which will be discussed further later, and in accordance with the ISO standard 10993 section 11. And we also need to be conscientious about environmental mechanical stresses. So we need to think about the material degradation and the material tolerances within the body.
So looking at some ASTM standards, we will be looking at some of our mechanical in vitro testing, starting at the top with a finite element analysis to analyze design parameters and behaviors of our controlled expansion. And then with coating inspection and acute particle characterization of our stent will have to be met, as our drug-eluting system will result in the release of several particles in the bloodstream. To do this, we'll be using scanning electron microscopy to analyze our device and to gain more knowledge into elemental and morphological data. We would also need to test the durability of our product in several different ways, primarily bending, axial, and toroidal. And then looking at the securement of our device, we will be creating an angioplastic model to represent how our stent and delivery system works, using a guiding catheter and wire surrounded by a 37-degree environment. So looking at our in vivo animal testing, first with our small animal model, we will be using a rabbit, as the iliac artery of a rabbit is known to be a reliable small animal model in stent testing. It's small and handled easily as it is a relatively inexpensive option. A rabbit model is also known to reflect human restenosis well, which is what our device aims to improve on. Traditionally, a pig's coronary artery has been the gold standard for animal testing, so we've chosen it as our large animal trial. This is because a pig's coronary artery is a good indicator of the safety of the stent, while giving a good insight of the biology and biomechanical properties reflected in humans. Pig hearts are similar in structures than humans, and several, around 10 pigs, will be collected to complete our trial. The animals will weigh between 30 and 50 kilograms, as the heart and coronary artery is of a comparable size of humans. For our animal tests, several time points will be taken on the small animal trials. They'll be taken after an hour, a day, and 3 days. And for our large animal grafts, they'll be looked at after 3 days and 28 days. So a considerable limitation to these animal models, though, is that they may not be a realistic model of coronary artery disease, but this may be fixed using animals that have a metabolic syndrome, and our animals will be held in a high-fat diet for the weeks leading up to the experiments.
So potential problems regarding our project. So an overarching problem is that the widely studied novel endothelial designs do not always address issues with the current DES novel stents because the scope of this analysis does not really include full clinical studies. Another problem is that patients with cardiovascular disease often have poor and dysfunctional EPCs, known as impaired progenitor cell activity. This can in turn lead to the dysfunctional reendothelialization when trying to capture EPC cells that are circulating or in the immediate area, which can limit reendothelialization. Fibrotic encapsulation is another potential issue, as is the formation of scar tissue on the surface of our device from myofibroblasts and fibrocytes. The tissue response to implants is in part dependent upon the extent of the injury created from the implementation of the stent and also the amount of provisional matrix needed for recovery. Current bioengineered stents do not outperform drug-eluting stents overall at the moment. Several trials have been done showing little outperformance in containing late lumen loss, which is directly correlated to binary restenosis. Drug-eluting stents again show occurrences of restenosis from 7 to 13% in stents. Stenting failure in epithelial recruiting stents will still occur along with thrombosis and restenosis, regardless of the excellent safety and efficiency of the newest devices. Therefore, new materials of both stent platforms and polymer coatings must be investigated to better the stent designs in the future. So the cost procedures vary from place to place, with hospitals being better equipped than others and costing more money. The variation in prices and procedures changes from patient to patient basis, with some having insurance and different other outside factors. The use of drug-eluting stents in primary percutaneous coronary intervention has shown benefits over bare metal stents in decreasing adverse cardiac events. For percutaneous coronary intervention, the procedure of angioplasty can range from $9,000 or $15,000 to $90,000. This total cost included further needed healthcare for patients that required extended medical attention. Depending on patient cases, there will be choices for several stents for angioplasty, currently between bare metal stents or drug-eluting stents. Drug-eluting stents have been found to have reduced restenosis and revascularization rates compared to bare metal stents, though bare metal stents can be preferred if the patient is no longer able to endure long sessions of dual antiplatelet therapy. Some drug-eluting stent examples are given with their respective prices, being the sirolimus eluting stent and also the Paklik axle eluting stent, which have been both significantly studied and documented. Despite the higher cost of drug-eluting stents, fewer reinterventions can lead to huge savings by the patient. Moreover, a higher patient's risk of reintervention, the higher the savings associated with the use of a drug-eluting stent. Additional costs can come from the further needed healthcare in the event of in-stent restenosis or stent thrombosis. Treatment of these can consist of repeat stenting using bare metal stent, a drug-eluting stent, or drug-coated balloons, again requiring further coronary intervention. Implementation of more than 2 metal stents in repeated in-stent restenosis lesions is likely to have detrimental effects on long-term outcomes of the patient, even though newer drug-eluting stents may improve the treatment of in-stent restenosis. If a patient develops restenosis after 2 or more layers of these stents, radiation treatment or brachytherapy is an excellent option for preventing another reoccurrence of blockage due to scar tissue. Thank you. Any questions?